Right, we'll go ahead and get started. Welcome everyone to the newest webinar in the GFRA Research Webinar Series with Dr. Antonello DiNardo today. Just a few quick slides before we get started. If you are not familiar with Zoom webinar, we will be using the question and answer feature. So that's the Q&A box that's down at the bottom of your screen. You can use this throughout the webinar to ask questions about our speaker's presentation, about his work and his research. We will have a moderated Q&A session after the presentation. You can also use this box for any technical questions and difficulties that come up throughout the webinar, as all of our panelists and moderators are able to see this, and we will be able to address your questions and concerns as they come up. With that, I will pass it off, and we can get started.
Hello, everyone. Good morning, good afternoon, good evening. Thank you so much for taking the time to join this GFRA webinar. Today, we are privileged to have Dr. Antonello DiNardo present on OpenFMD, which is a novel data sharing and decision support platform to enhance global epidemiological intelligence of foot and mouth disease. Dr. DiNardo is a senior molecular epidemiologist of the Vesicular Disease Reference Laboratory at the Pirbright Institute. His research revolves around multi-scale processes that drive transmission dynamics of infectious diseases, and by implementing models and methods adopted from molecular evolution, epidemiology, population, and ecosystem ecology to understand how viruses spread, change, and affect animal health. He is passionate about integrating digital technology with science, including novel options for global disease surveillance. Dr. DiNardo, welcome. Thank you so much for accepting the GFRA's invitation for this webinar, and we really look forward to learning all about OpenFMD. So I'll hand over to you. Thank you.
Thanks, Melanie, for the presentation, and a good morning, afternoon, and evening to everyone who is attending to the seminar. I'm going to share presentation. Okay, everyone can see that. Right, okay. What I'm going to present today is some sort of work project that I've been involved with a lot of collaborators in the last few years, and is the target, the aim of the entire project is to try to build up a new platform for enhancing the global surveillance of foot and mouth disease using new technologies, and in this case, digital technology, to try to support, as I said, the global epidemiological intelligence of foot and mouth disease. But actually, what they are talking about if we refer to epidemiological intelligence? If we look at what WHO tells us about epidemic intelligence, it is more specific than epidemiological intelligence. Is a framework that can be used for any action that can be targeted for the collection, analysis, interpretation of source of data from surveillance that enable us to understand risk posed by new pathogen threats, for example. And of course, making further action out of those informed decision and informed analytical tasks. The important things about epidemiological intelligence and any kind of surveillance activity is actually the data, because the data is crucial to get us know about disease threats, trends, and whatever is the new trends of risk of any new emerging pathogen or actually endemic pathogen as well. And there are different kinds of data that we can collect through surveillance activity. And all of this data inform us a different kind of level of resolution level of epidemiology like, for example, metadata collected through outbreak investigation can inform us about transmission dynamics and connectivity between farm. On top of that, if you move up to the higher level of resolution, we can track the spread of viruses, for example, of pathogen at regional global level through, for example, genomic data. And at the lowest level of resolution, we can understand, for example, mutation, how through the analysis of genomic data, how pathogen mutate and how these mutation affect the evolution. And all of these kind of data, all of the action that we can, all the data and the analysis that we infer from the analysis of this data is actually informing what we have to target about disease control, and specifically to control the disease and to reduce the spread of the disease in a more global scale, if we consider pandemic, for example. And so if you talk about surveillance data, one of the most important one is, of course, genomic data. And we have seen in the last 10, 20, 50 years, there is a lot of advance done in the field of genomics that has been quite influential in both reducing the cost and the timing of producing sequences, but as well in that sense, it is exponentially increased the amount of data that has been generated through sequencing through the use of new sequencing technology and sequencing platform. For example, if you look at the bottom left plot, there is a plot of the cost per human genome of a sequence in a function of the time of the last 20 years when sequences has been done. And if you see, in the last 10 years, there is a lot of reduced amount of cost that we actually pay now for generating a single human genome. And actually, this is lower than the Moore's law. If one knows about the Moore's law, is actually doubling time of the number of chips that is present in a CPU every 2 years. And this is quite hugely advanced, the genomics, instead of the Moore's law. And on the right-hand side, there is the cost of gigabase that we produce out of the sequencing as a function of the cost of the platform that we use. And for example, if you consider a platform like very highly costly one, we can reduce the cost of a gigabase up to, for example, $1. That is quite low, but instead we have to pay in advance for the sequencing technology. But if you consider a platform like very cheap platform like Mini-Ion, but they are quite useful as well. The cost per gigabyte increases a lot, but not that much. Is in the region of $10 to $100 per gigabase. And considering this kind of technological improvement of on sequences, we know we have a lot of data to analyze, a lot of data to share, a lot of data that we can use for informing control and surveillance of pathogens. And if specifically we look at the surveillance data, we have been witness during the last probably 2 years with the SARS-CoV-2 pandemic that the way we collect the data, the way we use the data has been changed a lot. So there is a lot of new technology that has been introduced by SARS-CoV-2, not only, but even before, that have moved from the normal standard way of not only sharing the data and the way we collect the data and with the way we use the data from the informal scientific publication form. So we publish data and we release data when the output is actually published in a peer-reviewed journal. But with the COVID, we have seen that there is a revolution in analysis and the sharing of information. So because the focus at that time was released in real-time data and analysis of real-time of the data to support control. And of course, sharing all these kind of data is an important component if we want to achieve a real-time surveillance of pathogen. As an example is in the bottom right side, is some sort of network, surveillance network that's been going on quite a while for COVID is the wastewater surveillance of COVID through different kind of partner. The network has creates some sort of an informal network where different kind of university, different kind of institution share their own data through different kind of dashboard to make this informed decision more available, more openly accessed by everyone. But this kind of idea of network is not really very recent. It has been going on for quite a while. Surveillance network has been existing for human pathogen, for seasonal influenza for quite a long time ago. And of course, one of the most important one in veterinary science, of course, the foot and mouth disease reference laboratory network that is actually led by the FAO World Reference Laboratory for FND that is here in Perplet and is facilitated through links with FAO and WOHA. In an essential is a quite an important institution that contributes to the global surveillance for FMD because it's on not only sharing data from actually field surveillance and generating epidemiological data, genomics data to understand the circulation and movement of the viruses across different regions of the world, different pools, FMD pool, but as well undertake a lot of other activity like vaccine selection and quality control, harmonization of diagnosis tests, and all the activity is actually focused on understanding more about the disease, but as well try to improve the surveillance of FMD throughout the activity that this is the task of the network. And importantly is that the FMD network has been seen like a good example that could be applied for other diseases, has been applied for other diseases of animal of interest of veterinary science, like for example African swine fever. As an example of the way data are shared through the network is the recent outbreak of the SAT2 topotype 14. It has been detected initially in Ethiopia in 1991, and in the recent year, in the last 2 years, has been quite an important diffusion and outbreak reported in the Near East, in Middle East, and especially in Iraq, in Jordan, Turkey, and Bahrain. And this is quite an interesting approach of the surveillance that the network does, and to understand the movement of viruses, emergence of a new threat in new region, introduction of new viruses into region, actually informing through the analysis of surveillance data for try to address in real time and swiftly the control of the disease. But as I said before, one thing that is actually missing in the activity of the network and as well in the surveillance for FMD is actually what we have seen and what we have witnessed for the COVID. So the use of new technology, for example, digital epidemiology, precision epidemiology for surveillance. So, and in a kind of way, the question is always how we empower the surveillance by using new technology. So by enabling, for example, research and policy through diversification and new analytical tools, and how we can make research and knowledge sharing more efficient through the use of this new digital technology. We have seen, for example, in COVID, the use of smart mobile phone for tracing of contact tracing of infection, or the application of machine learning, for example, or new way of visualizing the data both on the epidemiological scale and on the genomic side. So the idea behind this project I'm going to present you today is that we can try to create a new forum, a new platform to help everyone that is interested in the surveillance of FMD, but as well on the actionable results of providing control and providing information to ensure that we have appropriate control in place. how we can use digital surveillance for this kind of scope. And the idea is to, as I said, is to create some sort of forum, some sort of digital platform where we can not only increase the amount of data that we share, but as well try to create some sort of usable environment for harmonized data collection, but as well to provide people the ability to share their own data as well. And as well to try to create a digital interface for easy analysis of data that has been collected through in surveillance activity. So back in last year, well, more than 1 year ago, we had an idea and we created OpenFMD. OpenFMD is in itself is a website, is a portal that is how I can see that is a unique access point for all information, data, and analytics that we have for FMD. And in this case, so if you look at the website itself, is it just structured in such a way the user is guided through different kind of thematic section, as for example, genomic surveillance and vaccine topics, where the user can be guided through that and use different kind of tools and analytical tools, databases for not only retrieve the data and they're used by their own, but as well directly analyzing the data within the portal. The website and all the analytical interface that has been developed are actually hosted internally in the Perbrick Institute, managed by the WRL-FMD, World Reference Laboratory of FMD. I'm not pretty much going into detail to this kind of technological connectivity and specification of the web server. But one thing that I want to highlight here is that whatever we do with OpenFMD, we use open source environmental resources and software to deliver what we are achieving by OpenFMD. So we don't use any kind of licensing. We are actually quite reliant on open source resources. So as I said, OpenFMD is including a portfolio of interactive analytical tools that has been developed for different kind of tasks and different kind of use. At the moment, we have available 4 of them. One is still in development phase. FMD-Base is a database of the foot-and-mouth disease sequencing. FMD-Type is an analytical tool that can annotate and genotype new sequences that has been generated by everyone in the world. FMD-Watch is a kind of a surveillance resource for understanding burden of diseases and frequency of diseases across the different areas of the globe. And Pragmatist is a tool for that is pretty much target for vaccine bank manager or risk manager for vaccine selection in their own bank. I'm going to touch a little bit on all of those 4 in the next, let's say, 10 minutes. And I will start with FMD-Base. So as I said, FMD-Base is an curated database of FMDB sequences. It hosts the entire lot of sequences that is not only publicly available from GenBank, but is actually hosting as well private repository data that has been generated through the activity of the reference laboratory network of FMD. So at the moment is more than 18,000 sequences. We are around 19,000 sequences shared between public and private data. So the user can navigate across the database through filtering of different kind of metadata according to the virus lineages, country, host, collection date, and the type of genome region that the sequence is targeted to. There is the public and private data is accessible through different kind of, let's say, access level, registration level, while the public data can be easily accessed without any kind of registration. The private data require step-in registration, so the user need to register through to the portal, to the application, and abide to some sort of term and condition. This actually is the same one that has been going on for quite a while with the GSA data, GSA data with the seasonal influenza sequencing, and more recently with COVID. So from there, from either the public domain or the private domain of the database, the user can download their own data in a CSV format or a FASTA format. The CSV format provide further metadata that is associated to each of the sequences. And this application, this dashboard is open, is available now. So you can access from the www.fmdbase.org URL that is actually written in there. The next one, as I said, is FMD-type. So FMD-type is an analytical tool that is pretty much focused on generating genotyping report. Genotyping report is pretty much the one that we generate every time we receive some new samples, and we generate sequences in WRL, and we provide some sort of analytical tool for providing an information of which kind of lineage those sequences belong to, and generate a tree out of that. So is that kind of process. He moved to an open access tools, and user can friendly navigate through the tools and interrogate their own user data against the FMD database. So because the FMD type is linked to the FMD base, so it can access directly and can interrogate the entire database of foot and mouth disease sequences that we have in FMD base. So we can, of course, annotate the genomic sequences that the user input in the app. For moving to the reconstruction and phylogenetic trees, we extract the information from the VP1 coding region that the user can input. So the user can input even a complete full genome sequences, but the only one side of the only region that will be taken further for analytical purposes is the VP1 because it's the most variable one, and we use for genotyping and creating the report in the WRL. And you can, of course, create some sort of evolutionary scenario, because after generating the most likely, after pulling out from the database the most likely sequences that your user input sequence are related to, you can filter it out or include more that is actually pulling out from the database. The apps, of course, reconstruct phylogenetic tree that can be downloaded as a Nexus file and analyzed, further plot in using your own tools, for example. And the last thing that can do is generate PDF files as ad hoc genotyping report for your own purposes, or for generating some sort of surveillance activity. There's a little bit of video that is because the time of computing the tree is a little bit slow. It's not that slow, but it's taking, yeah, there you go. It's taking a little bit of time. So it's less than a minute. So it's 55 seconds, but as well, it's taking a little bit of time. So as shown in the video there is the interactive tree that is generated, so the user can annotate the tips according to, for example, the country of sampling, the year of sampling. They can annotate the tips as well according to the genotyping of each tips, for example, taxonomy level. And after that, the user, after few simple inputs of that is required for generating the report, like the country of origin of the sample, the date of collection, and the batch number, or if you want. The user can generate the PDF report using for their own purposes, as I said before. And the genotyping report comprise the most closely related viruses from prototypes and viruses for each of the sample that has been submitted, as well the three. moving on to the next one is FMD Watch. FMD Watch is actually a tool that has been developed for the main reason of epidemiological understanding the burden of the disease. Is actually pulling out the data from not only the database, so pretty much the metadata that is present for each sequence in the database, but as well pulling out the information from WAHIS, from MHOA, and integrate all these kind of source in a single repository to try to map events of disease in space and time, and to provide some sort of a geo-coordinate, geolocation of this point in a visualization form. So in a kind of ways, the aim of this map this app is to try to improve the time analysis communication of these activity, surveillance activity, to in real time update the database that is provided through FMD Watch. And as well, by understanding the burden of the disease, we can try to support evidence-based decision-making process for FMD control. There is not only the mapping environment for the application, but there is as well a plotting interface where you can follow the frequencies of serotypes or lineages through time to understand what is the actual burden of each of those serotypes or lineages within the region or within the country they are selecting. And of course, you can download the entire data set of data or filter it out according to your filtering for analysis in your own way or in your own computer. We have planned to expand a little bit FMD Watch because at the moment doesn't really hold or doesn't include the antigenic data, so vaccine matching data. One thing that we want to expand in the future, probably next year, this year, is to include the vaccine matching data in FMD Watch. The last one is Pragmatist. Pragmatist is a kind of a different tool because it's more target for vaccine bank manager, risk manager, as actually is a tool that enable the user to evaluate and prioritize candidate FMD vaccine strain for inclusion in antigen bank. So the idea behind this is the user can create and test some sort of risk scenario for region or country by parameterizing three main, let's say, quantitative parameter. One is the expert opinion of the risk that is perceived by the country for FMD coming or be introduced in that specific spatial resolution region of country. The prevalence of each lineages that you are targeting that could be introduced in your specific country, and as well the vaccine matching data that is actually already present in the application. It is generated from the data that is actually held within the WRL, but every parameter can be modified, it can be included, excluded according to the user need. The plots that is produced is actually a risk assessment of the high priority vaccine that need to be included in vaccine ban according to the likelihood of protection that those vaccine can confer according to the, as I said, risk scenario of region plus prevalence of lineages. In the future, we are actually thinking about to include a way of generating a report out of Pragmatist for users to use for their own purpose. So these are the main tools that are present at the moment, but we are continuing try to expand the portal to add a few more tools, few more analytical, a few more possibility for the user to interrogate those data. One of them is FMD Next, so will be the next train port for foot-and-mouth disease, and it will provide an entire understanding of the evolution of FMD by plotting tree in space and time, and will be pretty much seven different versions of an X-train, one for each serotypes, and it will generate some sort of the general evolutionary map of FMD in an application form. As I said, we will start by implementing the displaying of vaccine matching data in FMD Watch, like is something like in the bottom right on this slide. So in that way, the user can also visualize what is the vaccine matching data result has been generated, let's say, in the last 10 years, 20 years, from the well reference laboratory, for example, and what is the trend in time of the protection conferred by different vaccine against a particular lineage. And the next thing that we are thinking at the moment is to try to develop a module for the user to submit us surveillance data. So not only metadata generated from observation of cases of FMD in the field, but as well sequence data that is generated through different labs around the world. One thing that I want to point out here is that when we develop OpenFMD and all the application that is included in OpenFMD, those have been developed as a system that is generated by design. So that's mean that with simple and minimal modification, the entire system can be deployed and ported for other diseases like ASF, for example, or other diseases where a substantial amount of both epidemiological and sequence data is available and will be, of course, of interest for the research community and as well the risk manager community. Right, so last slide is what is the vision actually of OpenFND, because we are developing all these kind of things, but what actually our remit, what actually our scope with all these tools? Well, three main things that we may thinking about is that we have responsibility to try to connect, strengthen, and innovate with this entire portal or entire forum of idea. The connect is actually try to provide an environment platform where more people are able to provide their own data, or the sharing of information is quite easy to digest, and there is some sort of practice for the process of sharing of all this data. So I try to connect different kind of type of information in one single place where all the people can freely access them. Strengthen because we try to create some sort of collaborative environment for enhancing the epidemiological intelligence and, of course, surveillance of FMD. And this is not only done by a single person or a single actor. Is actually a collaborative approach where multiple people, multiple institutions, and multiple entities can strengthen this kind of vision and this kind of scope of increasing the surveillance and epidemiological intelligence for FMD. And of course, we innovate because we try to create we try to apply new digital application, new digital technology to the scope of providing back to the user and back to the community, to the research community, not only the research community, international institution, these available data, these available resources for their own use, for easy and open use for them. And as I say, this is not a single actor approach or a single actor way of generating the entire things, but has been quite a long-standing collaborative effort by different kind of people. And I would especially thank to the work that App Interactive has put in that in the development of the visualization dashboard, for example, and in the support that they are giving us for providing this visualization interface for the user to be used, but as well specifically to people in Perbyte as well, like for example Chandana Tenakun is a senior bioinformatician in Perbyte, as pretty much with me was the main actor as well, that we implemented the entire technological system or the interface of the web server, plus all the bioinformatic pipeline that has been developed and has been actually working in behind the scene for making everything smooth and working in the right direction. But as well, we want to thank a lot the fundings, UFMD, DEFRA, and UKRI, the BBSRC, for helping us funding most of the part of this work, and so without which we have not been able to do anything. As I say, so if you have any query, comment, suggestion, bug reporting, support, contact us. That is the email address when you can use to reach us. And thank you very much for listening, and I'm happy to take in any question if there are. Thanks.
Okay, thank you so much, Antonello, for that comprehensive explanation about OpenFMD. Clearly, this is an incredible digital platform for epidemiology and surveillance for FMD, and a tool that I'm sure everyone is going to be very eager to use. So I think we can go on to questions now. Just a reminder to everyone to please post your questions in the Q&A box. But perhaps I could start with the first question. So these tools are heavily dependent upon a database. How will you ensure that it is continually up to date?
Right. So the database is, as I said, is hosted in here in Perbyte. So I'm talking about the sequence database. So every time we generate new data within the WRL or within the network, we make sure that the database is continually updated in real time. So we apply a new version of the database every time we generate new sequences. And along that, we include the metadata associated with the sequences as well. From the surveillance app point of view, when a new case of FMD is detected by WISE, for example, or is reported to WOHA, or a new sequence that is comprised of metadata is sent to us, we make sure that in real time also the database that's feeding to the FMD Watch, the surveillance application, is up to date and of course display the new information.
Okay, that's great. Giselle, over to you.
Yes, thank you, Antonello, for that great presentation. So we have the next question is, the vaccine matching data is pragmatic, is heavily skewed to vaccines used by international vaccine banks, yet the FMD vaccines deployed in endemic settings are often locally produced. How can pragmatics be updated to include data for these other vaccines?
Yes, yeah. I forgot to mention that the vaccine matching data that is actually present in Pragmatist at the moment is using the vaccine or the vaccine antigens, the antigen that we use internally in Perbyte in the WRL for generating the vaccine matching data. Of course, this is not a comprehensive list of all the vaccines that exist in the world and the country uses in the field. But as I mentioned during the presentation, Pragmatist is heavily editable, so you can edit whatever you want according to risk region parameter, the prevalence parameter, and as well the vaccine data that is inputted in the app. So you can remove vaccine strain with vaccine matching data accordingly. and you can add your own vaccine strain with the vaccine matching data accordingly if you have it in your own lab, and try to create some sort of different scenario from what is defaulting to the HAP. One thing that I want to point out is that Pragmatist at the moment is specifically focused for an epidemic scenario. It's not really focusing on an endemic setting, while we starting to thinking about to update or create a new app that will use some sort of endemic scenario for evaluating the protection conferred by the vaccine. And in that case, we'll be much more, let's say, flexible in providing the any kind of vaccine data that the user has in their own lab, for example.
Thank you. Melanie?
Okay, the next question is from Mariano Perez. He thanks you for the presentation and wants to know, how far do you think we are from mastering the relationship between a particular viral genomic sequence and the actual antigenicity or antigenic features of the resulting viral particle?
All right. Okay. So as that's the $1,000,000 question. Not sure. Probably is more acknowledged than me, but on the digital, let's say, digital technology advance, we are starting to work. Probably starting from this year, we will have a PhD project starting in Pilbret in collaboration with Glasgow that will look at actually a way of using predictive models that has been applied for seasonal influenza to try to understand more about antigenicity and the connection between genetic and antigenic data in itself. With the hope we can try to dissect more about this kind of important thematic for FMD and try to understand if we can a little bit predict what will be the antigenic fitness of the virus according to their genetic makeup. But yeah, so it's a little bit of a $1,000,000 question, I would say.
Thank you. Giselle?
Okay, the next question is from Julam Gerol. What about private sequences? Will they be available in the phylogenetic trees? And they thank you for the presentation.
Okay, so FND type, so the application that is actually generating genotype report and phylogenies, doesn't provide any user the ability of downloading sequence data. Okay, it's just a tool for generating trees. But in the tool, all the data, both public and private, are pulled out from the database for the ability of the tool to create phylogenies out of that. So everything is available for the user, but it cannot be downloaded for sure. In FND base, as I said, so the database in itself, the private section of the dashboard that require the user to be registered, it will allow the user to download the data. But of course, there is a term and condition and some sort of equitable sharing of data policy that we implemented for not avoid the misuse of the data, but actually to control the fact that the data has been generated by other people and it be rightfully acknowledged in some way. And it's the same things that has been implemented in GSAID for years and years now. So everyone knows about that. So hopefully this answered the question.
Okay, in the sense of the private sequences that are submitted, is there a way that the sequences are screened or authenticated beforehand, before it is now free to be used in the database? Or is just a trust issue that we just trust the sequences are correct?
Now, every sequence that we include in the database is heavily curated by nick nodes. we should be 100% sure that all the data are perfectly in line with what we think. And if the data that has been submitted or the data that we even pull out from a gene bank is not of the right quality, or the metadata are not correct, we try to correct. If the sequence is not of good quality, we try to not include in the database. Right. So we have a process in place for quality control and curation of the entire database. Thanks to Nick. Yeah.
Okay. So if I have sequence data that haven't published yet and want to submit, then I just go through the registration process as you indicated, and then my sequences.
Now, probably maybe we have a little bit of misunderstanding. No, the registration process is just for accessing the private section of the database. For submitting the data, at the moment there is no direct submission. We will work on that in this year, next years. But at the moment, if someone want to submit any sequences, he can either use the email address that I presentation, or can contact Nick, for example. That's the best way to do it.
Okay, perfect. Over to you, Giselle.
So I guess you answer probably this question, but this person is asking, how can one propose a new tool to include in the platform?
Email address. I'm happy, 100% happy to listen to any comment, suggestion, or any new tools that the user want to include. And of course, regardless of our budget constraint, we can follow up. And even if people want to support development, they are very welcome to contact us.
So in the evaluation or testing for evolutionary changes, did you also look at recombination, and did you find any interesting recombination hotspots in the places where FMD is endemic?
Well, with the application I presented, there is no way of look at recombination because we haven't implemented any analytical regarding to that specific genomic task. But yeah, as everyone knows, there is a lot of recombination going on for FMD, but we don't specifically address that analytical task with our app.
Okay,
in general, when the tree is derived from FMD type, should be sure that is, well, not always 100% sure, but we should be sure that is 100% recombination free because it's just VP1. So
thank you.
So when you are using the OpenFMD and it's generating the phylogenetic data, you say there's a way that we could reference this platform?
You can use the URL of the website, but the plan is from later this has been submitted the paper, and so the plan is to have the manuscript published later this year, so everyone can reference the correctly when using the application.
Okay. Well, we don't have any further questions for you, Antonello. I think you have answered it all, and it was a really interesting webinar. We thank you so much for your time and your efforts, and it's been really interesting to see this new platform. I think you guys have really thought of everything in this digital technology here, and I myself am looking forward to using it. So it's very exciting. And thank you everyone for joining us for this webinar. I hope you have a good day or evening or afternoon further. Thank you, everyone. Over to you, Sheila.
Thank you. Thank you so much.
Thank you. I'll echo Melanie here and say thank you, Antonello, for the wonderful presentation and Q&A. And thank you all for attending. You'll see as you leave the webinar, a small survey should pop up. If you can take just a minute or two to fill that out, that would be really helpful so that we can continue improving on this series. With that, I hope everyone has a great day and be on the lookout for news about our upcoming webinars.
Thank you. Bye.
Thank you. Bye, everyone.